So good morning all. Thank you so much for the invitation, and I'm happy to share my thoughts here. And I also happy to see Dr. Kripanathi sir here. I'm being associated with him since 2008 when he was with Satya Sai University. So happy to see him here. Um, Today I'll be just giving an overview mostly because I'm sure that many of you have already gone through many papers, which is preprints, non-peer reviewed, peer reviewed, all those categories. So here we are trying to look at what are the key points that we have to consider on drug discovery and what are the possible targets. So here we are trying to analyze the drug targets of SARS-CoV-2. This is just a snapshot that I've taken a few hours back. This is a situation of the world with COVID cases, and the confirmed cases as per WHO in India is around 604,000 confirmed cases and more than close to 18,000 deaths. That's where we are reaching to, and there are bad situations still prevailing in many other countries. Hopefully, we'll come out of it soon. The terms and names, I have seen some of the presentations yesterday already. A few speakers have already gone through it. Might be a repetition. Please excuse me for that. The naming authority, I have given it on the left-hand side, and you can see the virus species is MERS and SARS, and the virus names have been given as CoV-2 for the new coronavirus.
Yeah.
Okay. Again, repeating it, here goes. So this is the current situation taken from WHO with the cases. So we are all working towards it, putting our brainstorming and putting our minds to it in all possible ways. The outbreak of this disease also probably started from a single or multiple zoonotic transmission events from the wet market in Wuhan, where meat and game animals were sold, and this was taken from the article Rio. And as during 2020, he has published, like early 2020, and a variant caused severe acute respiratory syndrome. And around 2003 later, where which is from camels, that's where the MERS came, and then December 2019, as a cluster of pneumonia cases were reported in Wuhan, where in China also reported where the SARS-CoV-2 came in. So this is where we have to have a clear idea. The disease is COVID-19, the virus is CoV-2, and the species is SARS. Now, the origin of all the different related respiratory syndrome viruses or diseases are starting from bats, then transferred to animals like civet cat, camel, and pangolins. And then there is also some relationship and similarity with the receptors when we are comparing with SARS-CoV and SARS-CoV-2. And they both have started during winter, and with the small cases, they got evolved. And this is something, it is being taken during Jan. I know the situation has completely changed as of today with the number of countries and deaths and cases. So please bear with me, these numbers are shown on the screen. So the attachment receptor is AC2 for both of them, but and also they expect TMPRSS2. There are a lot of studies going on in that, and the neutralization efficiency, it's being looked at high on CoV, and CoV2 is moderate. But again, now we are still trying to find out the right solution. So it is a single and a positive stranded RNA virus that belongs to the order Nidovirale and family Coronaviridae. And in March 11, 2020, the outbreak was officially declared as a pandemic by WHO. Its genomic size is from 29 to 32 kilobases, and CoV2 showed around 89.1% of nucleotide identity with bat SARS like CoV. So this identity is actually helping us as well as concerning us, because selectivity is a big criteria here. Then predicted replicase of open reading frames 1AB gene of WHCV is also in its length, and it also contains 16 predicted non-structural proteins, which is followed by 13 downstream ORFs. So these are all about the information about SARS-CoV-2 genome. And I am sure that yesterday you have already seen this picture or similar representation several times. So we are looking at spike proteins at different stages, different levels of targets. M proteins, hemoglobin esterase dimers, envelope protein, RNA and protein. So these are all being looked at several days. Now, coronavirus into the spotlight. This is what we are trying to compare between MERS-CoV entry and SARS-CoV entry. There are several studies being done on how about CoV2 on DPP4 as well as on ACE2. Both has been explored. Then only they came to a conclusion of ACE2 and TMPPRS. So here the entry mode, like it enters the target cell through an endosomal pathway, and then TMPRS2, which is an enzyme, helps the virion enter and virion releases RNA. So let me go take you through some of the steps, which you already know, but just to give a spotlight. So the spike protein binds to ACE2 receptor, then TMPRSS to the enzyme which helps to enter the virion, and then virion releases RNA inside the cell. The RNA translates to protein, right, and then replicates to make more RNA, and protein and RNA assemble into new virion in Golgi apparatus, and then it releases. That's how what we talk about virus release, then RNA replication and packaging, assembling and budding. All these stages are being taken care of. Yep. So finally, moving forward, this is the real way of attacking human cell. So you can see the cell and the virus that is getting affected on the cell. This is the real picture. It is taken from CDC. Now, here it's where I wanted to start more discussing about the targets. So here we're trying to compare between SARS-CoV and SARS-CoV-2. As you can see on the right-hand side, as you can see on the right hand side, both the viral strains, that is CoV urbani and CoV2 Wuhan 1, they have checked the percentage of identity, that is mean the structure, the particular strain on different ORF1AB, spike, envelope, M and N. So they did a comparative analysis and found a similarity there, as you can see on the right. This is taken from Trends in Microbiology, a very recent paper. And they also have found the respective crystal structures. In early Jan, we don't have much crystal structure. Most is big modeled, I mean homology modeled, predicted models. We had only 6LU7 at that time. Later, the group Diamond, they came into picture, and lots, hundreds of kind of, what to say, co-crystal ligand targets came in. I can see now it's about 82 crystal structures available in the PDB of different resolution, different conformations. Many more data is available. So the what we are trying to look at here is the main protease 3CL pro or non-structural proteins, that is NSP5, with unliganded active site is there in the pink color. NSP9 on the RNA binding protein, which is in the cyan color. As just to tell you the represent, the PDB IDs are also given here. NSP15, which is endoribonuclease, which is in marine blue. NSP16 and 10 is a complex in green, and perfusion spike glycoprotein in gray, and that is, which probably everybody knows, that is 6VSB. And I think a few others are also working on 6YB7 and 6VWW. Others, I know very specific groups look at it. Nucleocapsid protein N-terminal RNA binding domain, which is like RBD, is in the deep turquoise. So RdRp, RBD, these are the common terms that you usually hear when you here about the targets of SARS-CoV-2. These are the predicted models from I-TASSER. So this is like a genome-wide structure and functional modeling of SARS-CoV-2. Now, few of them are already available with the crystal structure, but still some of them you have to go for homology model structure only. So this is a really good snapshot. Please go to the I-TASSER website. You will find this data, and you can download all the PDB structures which is validated with safe servers and others being on these model structures on each stages. So the genomic organization is that's what is shown here. Each level cases, what are the corresponding like PDB IDs? This is what I have shown here. Just give me a moment. Yeah. So here what we are trying to see is that how like the RNA dependent RNA polymerase or endoribonuclease at which region and how we need to think of to be very selective. So when you're trying to do any drug discovery part of analysis, you need to be very specific on understanding the selectivity and sensitivity. It's not that we are doing for a multi-targeted thing. That's why even the what to say drug repurposing also it's taking a lot of time because of the selectivity part. So we need to be very sure that which is what kind of confirmation we are looking at, and then we also have to consider whether we are targeting on which mechanism, whether the entry, whether the replication or the release. So at each stages, it is actually a different kind of activity. So these are the possible drug targets that we are looking at. So the target names are given on the second column. The full name is like, as I told you in the previous slide, the papain like PL-PRO, 3CL-PRO, RdRp, that is RNA-dependent RNA polymerase, then viral spike glycoprotein, transmembrane protease serine 2, ACE2, AT2, which is angiotensin receptor. That is, and there are possible drug candidates also under clinical trials, and also suspecting that this could work or inhibit properly. The role of each of these target is when you're looking at 3CL-PRO, the protease for the proteolysis of the viral polyprotein into the functional units. That is the role here. And protease for the proteolysis of viral protein into functional is also for the PL-PRO. So PL-PRO and 3CL-PRO has a similar kind of role. Here, RdRp also tries to replicate with the viral genome and also the viral surface of the protein where the spike protein is looking at. TMPRS, as it is produced, the protease to primes the spike protein to facilitate into binding to ACE2. And ACE2 helps on the host cell, which binds to the viral spike protein. So for each stage, we have different drug being also looked at, the candidates being looked at. So that's where the targets here it is. And these are the just a snapshot of about the PDB IDs and its resolution, which gives you clarity about which crystal structure that could be taken for your molecular modeling or molecular dynamic studies that could be carried out. Mutations on the protein targets, nothing is being reported yet, but yes, on genomic variants, we found there are possible mutations studies, but yeah, nothing firm has been much reported. But as of today, if something is updated, I'm happy to know about it. Now, going to spike glycoprotein enclosed and open. So this is something that where modelers have to take care of. There are two different what to say in glycoprotein. So S1 is the host cell interaction. S2 domain, as you see on the picture, is involved in the trimerization. So the spike proteins, which are 3' are not much virulent, but we have to be very domain specific. So the open conformation, it moves the one domain from spike towards a receptor and interacts and hijacks. Therefore, endocytosis takes place. So the that's where the domain specific what I mentioned, including the conformation. So let me take you through a little bit more deeper into it. This shows the particular figure A shows the schematic architecture of the SARS-CoV-2 spike glycoprotein and also the degree of the protein surface conservation between the trimeric CoV2 and the CoV. So here also we have done a comparison between CoV and CoV2. A color range is shown in green and magenta represents not conserved and highly conserved respectively. The atomic positions of the cleavage site is indicated by an arrow. This is the atomic the site site. And coming to the this particular picture, the B. So here we are looking at more about other corresponding overload of AC2 receptor. So how AC2 is being interacting with the RBD of CoV2 as well as of CoV. CoV is represented in gray color and CoV2 on the blue color. Here you will see there is a difference in the particular loop. So the corresponding footprints are also colored, and to illustrate the overlap between both the interaction sites, this is where you can see this region. That's where they have similarity there. Now coming to C and D, here you will see the hidden RBD on the closed conformation, but on the open conformation, the RBD is being exposed. So this is the both the conformations are available in the PDB. So you have to choose the right one when you're going for any molecular drug discovery studies on the predictive side. Now, S1 to open to signals to enter the cells, as I already told you. This is also a paper which they have mentioned about the AC2 bound conformation at the first, second, and third conformation, then unbound up and unbound down conformations of SARS-CoV glycoprotein after trypsin cleavage and low pH treatment. As I am not sure how many of you are aware, pH has a great role in this binding of the spike protein or the cell into entering into the cell, the virus entering into the cell. So CTD1 is colored as a pink color, as you can see here, and the bound AC2 in the green color. The angle between the long axis of the CTD1 and horizontal plane is also shown at the bottom of each of these conformations. So this actually tells you about the role of pH treatment and trypsin cleavage. Even the bioassays also they try to study based upon the trypsin cleavage. There are some of the activity data available on the web. They have done assays on trypsin cleavage. Now, could DPP for, it's not DDP, sorry, it is DPP for inhibition could help or not? So as I told you earlier, they tried kind of a predictive models. It's not experimental. It's this particular paper refers to MD simulations and docking studies. I'm going a little more faster because the time is coming closer for the next speaker. So the glycosylation is also an important factor. So when they try to compare, the blue is more of the blue colored ones, it's more of a CoV related, and green ones are for CoV2. Now, what is this glycolysis to camouflage the virus? So it gets hidden from the some of the host antibodies. So these are the understanding of glycosylation site. If you can see for CoV1 and CoV2, they're closely related, but there is a difference. So that's where the 89.1% of identity and the balance difference makes this virus much more selective when compared to CoV1. And this is again arrangement which we are looking at the subunit level. So the mutations in the contact residues of SARS-CoV-2 spike protein, as you can see, these blue colored highlighted boxes, which is telling about the difference was aligned against the most closely related CoV-like coronavirus and CoV itself. So the key residues of the spike protein that make contact to AC2 are marked with the blue boxes, as I already mentioned, and in both CoV2 and related viruses. Therefore, the acquisition of the polybasic cleavage site and O-linked glycans, both the polybasic cleavage site and three adjacent predicted O-linked glycans are unique to CoV2. So this is where kind of a little bit of bioinformatics analysis being carried out to understand what are the possible mutations or what are the possible difference when compared to the other related respiratory syndrome diseases. Now, the structure of RdRp is also important. I'm not going too much deep into it because of time limit. COVID-19 NSP12 is that what we're looking at RdRp, and then NS5B and 3D pole for the comparison. So here you can see the NTP gets entered, which is a template entry, So there is a path where there are different positions, different features that to be considered on RdRp. So whoever working on a particular structure on NSP 12, 7, and 8, please take care of your domain and the interfaces and entry path. Then mechanism-driven inhibitors, as I already told you, at each level of stages or at each stages, you have to look at different inhibitors. So here, if you see, we are looking at virus receptor interaction, host protease inhibitors, fusion inhibitors, viral protease inhibitors, lipidomic reprogramming drugs that to be looked at. Then replication or transcription. So therefore, we are looking at nucleoside analogs and how to neutralize the virus. So at different stages, we are looking at to do drug discovery studies on how to inhibit them. And the drug targets and the possible inhibitors. I hope you know this picture earlier, which I showed you in the previous slide about the genomic organization. And these are the corresponding possible or under clinical trial, what are the inhibitors that is going on. Now, these are the few of the data that I have taken where the repurposed drug, they are going for COVID-19 trial, and it's a possible route of entry. Is also. So remdesivir is a prodrug which converts to GS441524 and inhibits the action of RNA polymerase. Galdesleukin, it's a viral RNA polymerase binder. And coming to Camostat, it's a transmembrane protease where people are all looking at TVMPRSS2, which is an inhibitor there. And flavipiravir is a prodrug, a selective viral RNA polymerase inhibitor. And hydroxychloroquine and chloroquine, we already have heard a lot about it, which inhibits the terminal glycosylation of AC2, and chloroquine also pretends to be there, including action of heme polymerase. Now, coming to Camostat, it's more of a COVID-19 treatment strategies, and also Camostat is looking at TMP-PRSS2 inhibitor and may inhibit the entry of SARS-CoV2 into lung cancer. So this is also taken from Cell Journal, one of the article recently published. So here also they looked at Camostat and nafamostat, what is their role and selectivity on different stages of the viral activity. Then remdesivir, of course, it's a broad spectrum of antiviral drug in the nucleotide analogs of adenosine. So here the mechanism by which terminates RNA chain was analyzed in this paper by also again RdRp, where they were trying to use Ebola virus. That's where it came from. And it has also a similar mechanism of RNA chain termination in SARS-CoV2. That's the reason why remdesivir it's also being tried for the testing. Now, just enthusiasts who wanted to start with CoV2 related molecular modeling studies on a very hit identification, not to design a lead molecule on a very initial phase, you could try with 6YB7, that is a PDB ID. This is the motif region where we are looking at the specific amino acids histidine 163 and globin 166 motif, and another catalytic site is a cysteine 145. So these two different amino acids have different role. I recommend you to please kindly read it and then understand where you wanted to dock and inhibit. It's not simply that the software is going to tell you this is the pocket and dock it. That doesn't make any sense. That's why I clearly mentioned what is a mechanistic driven drug discovery. You need to understand what is the role of that specific amino acid, how this amino acid is going to influence the mechanism or the pathway. If there is some sense, okay, then go and inhibit that particular amino acid. So non-covalent hits also over catalytic sites like cysteine 145 is also explored in 6YB7. Now, how are these therapeutics discovered? Organizers, I need two more minutes. I hope that's okay. Sorry for that. I'll be finishing it soon. So how the therapeutics are discovered? We have to identify the best biomolecular target which is tied to disease. And it should be very selective. And it is not that we are doing molecular studies or docking studies on a different PDB protein and we are doing entirely different bioassays. There should be some relation. So bioavailability, druggability, everything should be looked at the target. When you're looking at a hit identification, we are just looking a set of molecules which could interact with target. We are not finding an inhibitor. We are not finding a drug-like molecule. No, never. So just doing a screening or a docking analysis, we are just finding a hit molecule, right? Just doing a docking, you cannot get a drug molecule tomorrow on the table. Never, ever. So lead optimization is to be, that's where we look at modifying the hit with a better selectivity, non-toxicity, or many other parameters that to be considered. Now, when you're doing some docking analysis, it's not about only getting better score or energy. It's more about how about that particular geometry pose is can be crystallized on a biological complex. That is also important, right? So then later we go to pharmacokinetics and pharmacodynamic properties, how it reacts with the drug, how the organ also reacts of the body. So then metabolism, toxicity, everything will be taken care there. And animal testing, testing of efficacy in animals, and the process optimization where design, mechanism of manufacture, and scale of therapeutics. This is why I told the objectives of drug discovery is mainly to identify the compounds with an optimal balance of property. So by doing a docking or by doing a bioassay or getting IC50, you're just looking at this particular side, whether it is a hit or a lead molecule, and what is its potency, what is its binding affinity. That's all you're looking at. But by that, you cannot say that it is a drug-like molecule even, because in order to say drug-like, it should be safe. That means toxicity to be analyzed. What is the absorption rate in your body for that compound? What is the metabolic stability? Because if it metabolizes, when it reaches your target, of course, it will be different compound. So these are the metabolites, right? So we have to see whether we have to take a pro-drug strategy. What is the solubility? What is the permeability? What is the flexibility of the compound? All these parameters to be looked at after doing any kind of screening or hit identification. So that's where the objective of main drug discovery. Why I'm telling repeatedly, because many of us, including me, initial days, I thought molecular docking is just drug discovery. No. Molecular docking is a screening part where we are doing only hit identification, hit lead drug. In order to reach to drug like, there are tremendous amount of iterations to be done. So ultimately what we're looking at is, if you're not able to get such balance, then fail fast fails you. Drop the compound, work on the next one, and work only on the compounds or some modifications or derivatives only when you're confident, and avoid any missed opportunities so that you don't waste your time and money. So data overload is also a challenge, that you have too many compounds, and you have also 8 experimental assays, which you have a lot of 1,600 data points with you, or 10,000 compounds. You did several in silico studies and you have a lot of data points. But let me make it very simple. You have one compound with you, you did one experiment, sorry, 10 compounds with you, you did one experiment. It is easy for you to pick up the best 3 or 2 molecules. Now the same 10 compounds, I have 10 different experiments carried out. All 10 are important to me, independent, so I cannot take an average there. I have to consider all the 10 experiments data in order to choose the best 3 molecules among the 10. Now this is a bit challenging. Our usual way is we filter it from one by one. First experiment, then filter, second experiment, filter. But in drug discovery, we cannot do that, because sometimes it might be tightly binding, but it might be toxic. If you try to modify some regions of the compound, the toxicity goes off, still the activity will remain there. So that is called optimization, right? So because of this challenge, you have a lot of data from in silico, in vitro, and in vivo. Now we have to prioritize them. Which property is more important and which property is less important? For me, everything is important, and for us, it is always, but not like that. For oral driven or intravenous driven, you have different priority of PKPD or PK properties or pharmacokinetic properties that to be considered. So based on that, you have to prioritize them. So what is the importance and what is the uncertainty? And then you have to make a selection based on diversity and quality. Means it is the selection of the best hit molecule is not based only on the score or not based only on the energy. Never it is possible. There should be diversity. That's where we have to think about. We always do one time docking and we say we got the result. No, it is an iterative process. First, we have to consider more of diversity and then score. So let's say 75% of diversity, 25% based on the score, you select the top 10 or top 10%. And then on the next iterations of whatever you're doing, you give more priority to your score. That means 75% importance on score, 25% on diversity. That's a difference that we have to understand. That's how pharma works. So we have now we say we don't have money. Of course, drug discovery needs money. That is there. So that's why we have to understand on selection. That's the only reason why many of them blame that when you're docking and you're getting some data, when I do bioassay, I don't get the matching result. This is because your target might itself not, whatever taken for docking studies is not druggable or bioavailable. They have difference in the bioassay, and also you not consider diversity and quality in this. Now, what is the impact of computer-aided drug discovery on pharma? If you see, there are many compounds already in clinical trial or in phase 2 or phase 3 as listed here. I have taken from these reviews which are being shown at the bottom. Now, also let me tell you, using just one tool or one software, nobody is going to bring down a drug into the market. There is a combination of multiple tools to be used. Don't think that using a single tool you can get something on your hand. No, there should be combinations of multiple tools used. So that many tools are now freely available. Some are proprietary and paid and available. It all depends upon what is your priority. So when you're choosing some of the tools, it is very important that you test, evaluate all of them, and choose the best one for your problem. So for problem A, software A is good, but for the same software might not be good for problem B. So that you are the master to evaluate and understand that. For me, A might be good, and if I recommend you, but for your problem statement, that is not good. So we need to have a better understanding on what the kind of algorithms helps you to solve drug discovery on the computational aspects. Now, why no vaccines yet? Of course, currently many progress have been made, but I don't want to comment on that. That's why I didn't make any claim on COVID-19 vaccines here. So they're scaling up, and manufacturing is hard. That's why it is very difficult and it takes time. And short term is for repurposing the existing compounds. Let's see. There are some news going on. So let's see if something comes up. Now, the other resources or modelers on SARS-CoV-2 are these. I'm not spending too much time here, but whoever working on molecular dynamics, I don't want you to repeat again because you will say there is no hardware resources. So D. Shaw is a company who has done on microseconds, who are knowing about molecular dynamics, microseconds range, on most of the targets of COVID-19 or SARS-CoV-2, and you just download and analyze them. And the software for analyzing is called Desmond. Works on Linux. It's completely free for academics, so you don't pay a penny for molecular dynamics analysis there. Anyway, on PDB also you have a lot of resources available. So please go through them before doing any kind of molecular modeling studies. Just two marketing slides. So being I am a coordinator here for this particular national initiative from the government of India and MHRD, as well as organized by ASCT and CSR. So there will it is yesterday being officially launched, which is called Drug Discovery Hackathon. Anybody can take part. Students, faculties, researchers, even international participants also can take part. So please go to the website for more eligibility criteria and more requirements. The track one is completely looking at the virtual screening, pharmacophore, molecular modeling on COVID-19 related targets. Or track two is completely on tool development. If you know programming, Python, or if you have an enthusiastic computer science efforts, so you can do programming. And track three is completely moonshot track, which considers completely disruptive approaches. So I thought, being we are talking about COVID-19, why not we talk about this initiative also? Of course, there will be prices, and the prices all depends upon how good is your result. Keep in mind, diversity should be kept in mind, and quality also should be kept in mind. That's why I emphasized on that point. So many things will be. So it is good that, and I also request everybody, especially students, because it's a learning process. You learn many things through training. And some of our offerings from Shastra Innovations. So I don't put in any much things here. I don't want to do it as a marketing thing. So, but we take care of most of the solutions for machine learning based QSR, human toxicity endpoint predictions, ADME, and the bioisosteer replacement, who is a medicinal chemist, it will be useful. And if you're designing some new molecule, if you want to do some retrosynthesis pathway. So not taking much more time. So I'm thanking all our superheroes in the front for this fighting against COVID, and also the organizers of this conference. And I also thank Mahesh, who gave me an opportunity and invited to this particular platform. Thank you so much, Mahesh, from LLB. And also my student, Miss Ambli, and my other collaborators, Vijay and Sachita, and group of researchers. Thank you so much.